oxidizing with the light harvesting complex and with the oxygen evolving complex. The light harvesting complex is oxidized, which starts the ball rolling, starts the electrons rolling. And immediately then the oxygen evolving complex oxidizes water to produce oxygen and protons. And we can follow these protons through ATP production. And then we can actually follow the electrons, the excess electrons, through to reduce NADP to NADPH. Now, we go to the dark reaction summary. So here we've produced the NADPH and the ATP, and here is how they're used. This occurs in the stroma of the chloroplast, and we reduce these complex reactions down to 3 stages. Carboxylation, which is that one reaction in which RUBP, a 5-carbon intermediate, is combined with atmospheric CO2, or CO2 from the atmosphere, to produce that unstable 6-carbon intermediate, which breaks down to triose phosphate. Then the majority of this, the NADH and the ATP, is used in the reduction phase. Why is it called reduction? Oh, come on, folks. Why is it called reduction? It reduces. It goes through a series of reductions. So that compound is reduced to now what is a triose phosphate. And then the majority of the reactions and some of the ATP is used in regeneration to start off with the starting material. And it's a pretty complex reaction. You just need to know these 3 phases. You need to know when the light reaction products are used, which is in reduction for the most part, and a bit into regeneration. Okay? Now, let's pick back up on where we were the last time, where we discussed this. And you've seen this. I think you had this probably several times by now, and I told you would have it again, where in the real world, that enzyme Rubisco, which grabs the CO2 and the RUBP and converts it into this, adds a CO2 to convert it into the stable product, also uses oxygen as a substrate. About 1/4 of the time, maybe less, maybe a little bit more. So that if we increase the oxygen concentration, and this has been known for quite a while, then you actually see a reduction in the relative rate of CO2 uptake. And this is the PCO, or the photorespiration. It is called the glycolate cycle. Glycolate is the product. It's a C2. It is a 2-carbon compound, and it involves the chloroplast, the peroxisome, the mitochondria. The peroxisome I don't think we've covered before, and you just need to know those 3 organelles. You need to know the individual reactions. And basically, photorespiration, we end up with losing a CO2. So every time there's an oxygen is fixed, we lose a carbon. That's not good. We're going negative. And this really happens. It's not just something we do in the lab. This is going on outside right now. Now, we don't really know why this is happening. And now what we're going to talk about is the way plants have evolved to reduce this photorespiration, okay? And you've probably heard of these. These are the C4 plants. And then there's another kind of photosystem or dark reaction system, CAM. CAM is not nearly as efficient as C4. And we're going to spend some time talking about the setup for C4. It's called C4 because the first stable product is a 4-carbon organic acid. It's called C3 because the first stable product is a C3, 3-phosphoglyceric acid, a triphosphate. Pretty easy why they call them C3 and C4. Now, what we're going to talk about today is there's a lot of reasons why C4 plants are more efficient. There's not just one thing. It's just not photosynthesis. It's structure. It's where they're found. There's a lot of things that go into making the evolution of C4s much more efficient. In terms of plants, in terms of species of plants, C4s are not a large number of species of plants, but they're overly abundant when it comes to crops. Because when men first started going from being wanderers to starting to do agriculture, they're going to select crops that they could eat and that grow well, right? You know, so they were doing a lot of stuff thousands of years ago, selecting seeds that germinate and that produce something you can eat. And if it grows fast and it produces something you can eat, then you're going to start using that more and more. So they then started creating, you know, most of the major crop plants way before modern agriculture. You know, everything was, you know, domesticated and developed over thousands of years. Anybody know what the word Kranz means in German? Anybody speak German? No? No one speaks German? So I could just make up anything and you'd think it was right then. It means ring. I think it also means halo. And that's because the first thing about C4 plants is they have, and this is important, they have two totally different kinds of chloroplast. They have the bundle sheath cells, and then they have the which are right around the transfer system, the phloem, and then they have the more loosely packed normal mesophyll cells that you see in a normal C3. Now let's look at this, and I want to make some points here. Look at this. This is chloroplast in the bundle sheath. So right here are these chloroplasts. Here they are, and here they are, okay? And right here, anybody remember what this tissue is, this smaller tissue in here? What's this stuff? What's the general name we call that stuff there? Vascular. The vascular tissue, which is the xylem and the phloem transfers carbon out. So phloem is going to pull in sucrose and move it to the rest of the plant. So one point that people usually don't make is that the transport system is more efficient. You're making the sucrose, the plant is making sucrose right at the point of transfer. Okay? So that makes it more efficient. We have a different set of reactions that occur here than occur out here. Okay? So here we have a stomate, so the CO2 is going to come in, and to tell you the take-home message, it's first going to be fixed in these cells, in the mesophyll cells, and then the product of this first fixation, the C4 carbon is transferred to these cells where the light reactions happen again, and we have the dark reactions, okay? So you have the carbon fixed here first, and then it's refixed here. So how much energy does that require? compared to a normal C3 leaf that we've just talked about? A lot. Where do we find C4 plants? Closer to the sun. More toward the center of the earth, toward the equator, where there's a lot of sunlight, a lot of water. These plants use a lot of water. They're more efficient, but they use water. So they're evolving in an area where actually we first probably started evolving and starting to set up farms, okay? A lot of light. So there's a lot of things going for C4s. They're fixed here first, then they're fixed here again, okay? We have CO2 fixed in these cells. We actually have a decarboxylation reaction. We're going to go over these in detail. What's a decarboxylation? We break off a CO2, so we fix the CO2 here, move this 4-carbon compound into these cells where it is decarboxylated, and then it's refixed, okay? The cells out here do not have rubisco. So these cells are not inhibited by oxygen. Then we concentrate the CO2 here so that we outcompete the O2 back from enzymology, remember? Now, subtropical, 18 different families. You can't look at a dicot or a monocot and really tell what it is just by looking at it a lot of times. You can a lot of times, sometimes you can't. And they're also a little more resistant to drought because they're more efficient. The key thing here is that in those mesophyll cells, the enzyme that fixes CO2 is phosphoenolpyruvate carboxylase, PEP carboxylase, or pepcase. And this reaction, which occurs in those mesophyll cells, produces OAA, which is a 4-carbon organic acid. You should just know 4-carbon organic acid, okay? This enzyme, which fixes the CO2, does not see oxygen. Okay? So that enzyme does not have to worry about being inhibited by oxygen. So let's look at this cartoon. We have a mesophyll cell and we have a bundle sheath cell. Let's go back here. Mesophyll cells are out here. The bundle sheath cells are right here. So sometimes this is a little confusing because, you lose the location. So here, most of the cells, right here is a bundle sheath cell, then right here would be the phloem. So what happens? Let's start at CO2 entering the mesophyll cells. CO2 enters the mesophyll cells. Then the enzyme PEP carboxylase uses as one substrate phosphoenolpyruvate, PEP, and CO2 to produce then this 4-carbon oxaloacetic acid, OAA, a 4-carbon acid. This 4-carbon acid is reduced, and then the thing that you need to know is that 4-carbon acid then is transported into the bundle sheath cells. So now we have the acid, the C4 acid produced out here, and it's transferred into these cells. In these cells, what happens then is the 4-carbon acid is decarboxylated to produce CO2, and then that C3 acid then is transported back to the mesophyll cells to keep the cycle going. Got it? So CO2 is here, and CO2 now enters what's called the normal C3 cycle. Got it? Now what is happening here is this is going on and going on, and we increase the concentration of CO2 in these cells. So if oxygen is a competitive inhibitor of Rubisco, how can we overcome competitive inhibition? Nobody remembers? How can you overcome a competitive inhibitor?
The limit is increase the ratio. If you got literally nothing but CO2,
right,
then that's what it's going to go. That's what it's going to.
These cells still have oxygen in them, but what we're doing is we're increasing the concentration of CO2. So if we have the same amount of oxygen and we have a lot more CO2, what's the probability of an oxygen being fixed? Low, right? So what has evolved is this system of concentrating CO2. Rubisco is here. Rubisco still will bind oxygen. It's just a numbers game. It hardly ever, if ever, binds oxygen. So one thing we can say about C4 plants is the rate of photorespiration is extremely low. So we've had several things that happened over time. We have this enzyme that now fixes CO2, and this enzyme, which is out here, is not, oxygen is not a competitive inhibitor, remember? So we concentrate it. Here, it doesn't matter. In here, then this CO2 is going to continuously increase and increase, and this is going to then be fixed, and there's no photorespiration. And indeed, the photorespiration of a lot of C4 plants is not detectable. It's going to happen every once in a while, of course, but not very often, not a quarter of the time. Okay, questions? So you have two kinds of chloroplast. You have this double fixation, and then once the carbon is fixed in the C3 cycle, all plants have the C3 cycle. It's just that these plants also have another set of enzymes that will fix carbon, the C4 cycle. Then once the carbon is fixed, it's just ready to be transported. Okay? Questions? Okay? We've talked about this. We'll talk about it in summary. Needs a couple additional ATPs. That's okay. This happens where there's a lot of sunlight. And it's a couple of ATPs if you want to do the stoichiometry of the actual reactions. But these plants produce more carbon. They produce more sucrose per unit time. They're just more efficient. And then they're able to move that sucrose out quickly. So it's structure and its function. A lot of things had to develop for this to happen right. Okay? Questions? Okay, something we've not talked about is something called the CO2 compensation point. And I don't know if I put the curve in here or not. No. We can take a plant and we can put the plant in an environment with different concentrations of CO2. Okay? And this CO2 compensation point is if we put a plant in different concentrations of CO2, the more CO2, the more photosynthesis. Okay? But if we start taking the CO2 level down, we can get to the point of where whenever one CO2 is fixed, a CO2 is given off from respiration. So we take a plant, we put it in a container, and we lower the amount of CO2, and we keep lowering it till we get to the point of where it's balanced. So the plant's in light, and the concentration of CO2 is such that for every CO2 that's taken up by photosynthesis, 1 is given off from respiration. Does that make sense to you? So the CO2 compensation point for a C4 is very low. In fact, and we try to do this experiment, I try to do this for class, and you have to have a lot of light to do this. And I'll try to find this on YouTube. You can take the classic experiment is to take a C3 plant and a C4 plant and put them together in an airtight container with a lot of sunlight. The C4 will thrive. In fact, the C4 is robbing the C3 of carbon. Remember, the C3 is going to be giving off CO2 and taking up CO2. The C4 is going to be taking up CO2 and giving up CO2, but it takes up a lot more CO2. So you can actually see over time the C3 plant start to die, and the C4 plant's just fine. It is actually robbing the carbon from the C3 plant. It's a hard experiment to do in a growth chamber because we need a lot of sunlight. And you can seal them up, and they'll go for quite a while. Okay? They have C4 photosynthesis, has a higher temperature optimum. Quantum yield remains steady. C4 quantum yield decreases. That just means as C3 plants get hotter, what's a quantum? It's a measure of how many moles of light, if you want to do it that way. We see a breakdown in the actual efficiency. And then C4 plants also are more sensitive to low temperature. Why?
They evolve in regions that are like generally tropical.
Yeah, they evolved in regions that are not that cool. Why are there no C4 trees? Why are there no C4 trees? I don't have a clue. And I've not been able to find a C4 tree. I don't think there are any C4 trees. Oh, well, maybe some trees, and there's some other reasons, you know. C3 plants, and we'll talk a little bit about this probably, they will operate a little bit better at a lower light. So if you think about as you go upward from the equator, the quality and quantity of light starts to decrease, you know. So they're a little more efficient then. Okay. Here is quantum yield, where we look at two different kinds of plants. The red one is a C3, and the Atroplex, of course, is a C4. And as you see the leaf temperature increase, of course, you're seeing a reduction in the quantum, yield, quantum yield, CO2 uptake. And by the way, we actually measure moles of light. So we can actually measure, remember, light is it's a wave and a particle, so we can actually talk about moles of light. So we see a reduction in that, and we don't in the C4, okay? Okay, again, just to reiterate this, C4s are more efficient at really light-saturated conditions and at lower temperatures or lower amounts of light. C4 plants can be more efficient. I mean, C3s can be more efficient than C4. Okay, now let's look at some, let's compare these two. Photorespiration, C3, yes. C4, not detectable. And also the C4s don't have the organelle structure. So if you take a C3 leaf and look at it in an EM, you see the mitochondria, the peroxisome, and the chloroplast are just mashed up against each other for the C2 cycle, for photorespiration to occur. And that's why a lot of people think that photorespiration has some role that's important. because they're just pushed right together. Temperature optimums, photosynthesis, 2025, 30 to 45, Rubisco, C4, PEP, the temperature, the optimums, quantum yield, declining, steady, transpiration ratio. So this is a biggie, okay? These usually are found where it's well watered, but their transpiration ratio, in other words, water to CO2, is much lower. Light saturation, C4s do not saturate. So they're capable of, and I mean by light saturation, I mean we're talking about past normal amounts of light for photosynthesis. So we're not talking about, you know, as bright as the sun type light, but it just keeps becoming more and more, it keeps producing more and more carbon. Okay? Now let's switch gears. Crassulation and acid metabolism. Where do you find CAM plants? Where there's no water. Are plants that grow in the desert efficient? Do they grow rapidly? No, with some exceptions. There are some small plants, seeded plants in the desert that when they get a little bit of water, they germinate and go like crazy. And then they're finished in a very short period of time to produce the next generation of seed. But for the most part, these plants we're talking about do not grow very quickly, and they have evolved to live in conditions where water is an important factor. They're also found in plants that we call succulent plants, and there are even plants that are C4 CAM. They can switch back and forth between being a C4 or being a CAM, depending upon where they're located. Now, the difference between a C4, a CAM plant and a C3 plant is that a CAM plant has the two kinds of photosynthesis, has the C4 and the C3 kind of photosynthesis. But we have one system, the C4 happening in the light, and then we have the C3 happening in the dark. So they are divided up by time, and they don't have any kind of specialized chloroplasts for the most part. So, flip up to... So what happens then is that during the day or during the night, the stomates are open. And if you look at a stomate in a cactus, it's not like a normal stomate. It's usually down inside a hole to decrease water loss. So now we're running into the problem of the only way we can get minerals into the plant is through the transpiration stream. But we want to decrease the water loss but still allow CO2 in. So stomates are open at night, and the CO2 enters through the stomates and is fixed as a 4-carbon acid, just like in CAM plants. Then the stomates are closed during the day. So at night, the plants accumulate the 4-carbon malic acid. Okay? Then during the day, the malate is transported, decarboxylated in the cytosol, and then the CO2 enters the chloroplast and is fixed in the normal C3 cycle. Okay? The C3 cycle requires light reaction products to work. So during the day, the plant is producing NADPH and ATP, which is dumped into the stroma of the chloroplast. The CO2 enters the chloroplast and it's fixed. Okay? So we have a night versus day. At night, the CO2 enters the plant when water loss is at a minimum, and then in the day, it's fixed. Again, we have two fixations. And we can look at the cartoon here. Here we have CO2 uptake. Okay, this is dark, this is light, so we see this CO2 uptake at night when the stomates are open, and then it just goes down very rapidly, and in the light, it's much, much lower. That's because the stomate is closed, so we're not losing water, and we're not uptaking CO2 at night. And in the desert, temperatures are very, very low. So the loss of water, and a lot of water loss is through the thermal agitation, right? If the leaf is hot, it's going to lose more water just because the molecules are bouncing around, right? So at night the CO2 enters, CO2 enters, water is lost, and then the CO2 then is reduced or is carboxylated to produce malic acid, which is stored in the vacuole. This is the same cell at night. Then at night, the malic acid then is moved into the cytoplasm where the decarboxylation occurs. This is also a difference. Everything else we've talked about has happened inside the chloroplast. Now we have something happening in the cytoplasm. And then the CO2 enters the normal, I'll call it the normal C3 cycle. Okay? Yes, ma'am. The ATP and then ADPH is produced in the light in the chloroplast. Okay? So for these reactions to occur, these reactions occur in the cytoplasm, and they use other reducing compounds. Okay? No ATP, no NADPH without light. Starch is actually stored, it's broken down to triose phosphate, and we're going to talk about movement through the movement of the translocates probably next time we're not meeting. And then during the day, we have ATP and NADPH being produced, which then reduces the CO2. So now we have night versus day, and also now we have more of an involvement of the cytoplasm. These other reactions occur, let me go ahead and go back up here, in the chloroplast. Okay? Questions? Day versus night. So if I give you a plant in the and if I give you a plant in a box, this is a common question I ask. I give you a plant inside a box. It's either a C3, RC4, or a CAM. How can you tell what kind of plant's in the box? You know, and you can do everything but look at it. Photosynthetic efficiency, or you can grind it up, you just can't look at it. So does it have PEP carboxylase? Is the decarboxylation occurring in the cytoplasm? There's a lot of things that you can use, you know, in these sort of critical thinking type things. Okay? Both have PEP carboxylase and both concentrate CO2, but in CAM plants, the carboxylation and PCR cycle are going to occur in the same cell. Okay? CAM plants have incredibly low transpiration rates because they don't have any water to start with. Remember, we're going way back to this conundrum of the only way that the minerals that are required for enzymes to work can get into wherever as high the plant is by moving up with the transpiration stream. Also, we know desert plants have evolved to not have leaves or have very small leaves. There's not just one thing. There's a lot of things that go into those plants thriving in the desert, okay? Questions? No questions? Okay, we'll stop there and we'll pick up on translocation next time then. I'll be around this afternoon. I'll send out an email if you want to. Nobody showed up for the help session yesterday. There'll be one at 6 and 518, which will also should be broadcast live. If you need to talk to me online or whatever, just either send me a request or go that schedule one thing and schedule a time and I can be there, okay? I'll send that out to everybody. I'm sorry? 518 is when you get off the 5th floor of the conference room.
I did kind of a cool experiment a few years ago. I tested like a series of plants that, between C3, C4, and CAM, really just the difference between C4 and CAM.